Welcome back to the deep dive. We're the place that, cuts through all that noise, does the sifting for you, and brings you those insights you actually need.
Insights to be truly informed.
Exactly. And today, we're tackling something huge. A challenge that's, well, it's been around forever, but keeps finding new ways to outsmart us. Cancer.
Right.
We all know how cunning these cancer cells are, don't we? They're not just, surviving. They're described as metabolic tacticians.
That's a great term for it.
Constantly rerouting fuel, rewriting their own rules. Honestly, often outpacing our best treatments. It's frustrating.
It really is.
But what if we could totally flip that script? Achieve what's being called metabolic sovereignty over these cells. Dictate the terms instead of just reacting.
That's the core idea we're exploring today.
Yeah, it's this really audacious concept from a pretty groundbreaking article titled Nanomachines of Collapsed Dual-Layered Protocols for Metabolic Sovereignty.
Quite a title.
It is. It's by Saikou Venga, also known as Dr. Melvin Sewell, published in, and I love this name, Bibebibebibe Magazine.
Bibebibebibe Magazine, okay. Definitely sounds like it pushes boundaries.
Doesn't it just... So this article isn't just suggesting another therapy.
No, definitely not. It's proposing a fundamentally different way to think about cancer metabolism. It argues that the very adaptability of cancer, its biggest strength, actually leaves behind a metabolic signature.
A signature. Okay.
Exactly. And this new tech aims to read and exploit that signature using these dual-layered nanomachines to basically collapse the whole operation from the inside out. No escape route.
Wow. Okay, so that's our mission for you today. Let's unpack these really ingenious dual-layered nanomachines. Let's figure out how they supposedly deliver a checkmate in this metabolic chess game against cancer.
It's a fascinating concept.
It really is. Get ready, because this could be a glimpse into a whole new way of tackling this disease. All right, let's dig in. The article really hammers home just how good tumor cells are at surviving. Not just resilient, but actual metabolic tacticians.
It's a key point.
Think about it: constantly switching fuels, rewriting enzyme hierarchies, finding new energy tricks, adapting faster than we can often react. It's like they have this whole playbook ready.
Always one step ahead, changing the game plan. But, and this is crucial, while that adaptability is a massive challenge, it's also their potential weakness.
How so?
Because every adaptation, every metabolic shift leaves that trace we mentioned, the signature. It's a pattern. And the article mentions this tool, the reality level analyzer, designed specifically to sort of detect and understand these signatures.
Okay, the reality level analyzer.
Yeah, it's supposedly key for designing these nanomachines. It lets us get ahead of the curve, not just chase their latest adaptation. It's about understanding their core strategies.
So it's predictive, almost.
That seems to be the idea. And connecting this back, the ultimate goal isn't just, poking holes here and there. It's architecting devices that can systematically collapse the whole metabolic infrastructure.
From the surface right down to the core.
Exactly. From the cell surface, how it interacts outside, down to its deepest energy processes. A really comprehensive takedown, not just a localized strike.
That makes sense. And that brings us to the main strategy, these dual-layered nanomachines. The article gets pretty dramatic, saying they're designed to breach the glycolytic sanctum and sever the tumor's energy rituals.
Yeah, energy rituals. It's vivid language.
It really is, like nanotechnology meets ancient prophecy.
Yeah.
But the clever part you mentioned is they both start by targeting the same things on the cell surface.
Right. They share initial targets.
Things like HER2, we hear about that with breast cancer often, PSMA for prostate cancer, and EGFR, common in lung cancer. The article says targeting these ensures selective binding to high-density tumor terrain.
Which is just a smart way of saying they lock on to where the cancer cells are most concentrated.
Minimizing collateral damage, presumably?
Precisely. Targeting HER2, PSMA, EGFR is absolutely key for that precise delivery. These receptors are often way more abundant on aggressive tumor cells compared to healthy ones. They act like beacons.
Guiding the nanomachines in.
Exactly. It allows for really specific delivery, minimizing off-target effects, and using this two-pronged attack, both machines using the same initial guidance system, it aims for this comprehensive assault right from the start. Overwhelm its ability to adapt.
Okay, let's break down the first wave then. Nanomachine first. It focuses on the enzymatic collapse protocol. Sounds pretty direct. How does it work?
Right. Nanomachine I is like a multi-stage weapon. Layer 1 is that surface receptor targeting HER2, PSMA, EGFR. That's the lock-on.
Got it. Binds to the tumor cell.
Yes. Then layer 2 kicks in. Glycolytic pathway recognition. And this is smart. It's not just binding, it's diagnosing the cell's metabolic state.
Diagnosing how?
It looks for specific markers, things like GLUT1, that glucose transporter that cancer cells often overuse, and key enzymes like hexokinase II and LDHA. These are all telltale signs that the cell is deep into Warburg metabolism.
Ah, the Warburg effect, that rapid energy production even with oxygen around.
Exactly. So layer 2 confirms, yes, the cell is relying heavily on that fast-burn glycolysis.
All right, this is where it gets really interesting. So it binds, then it confirms the specific metabolism is active. That's seriously intelligent targeting. And then it unleashes its payload, the dual enzyme disruption.
That's the punchline.
And it hits two enzymes, not just one. Why two? What does that achieve?
That's the ingenious part. It goes after two absolute linchpins in that glycolytic pathway. First, pyruvate kinase, specifically PKM2, the version often found in cancer. Think of PKM2 as the main tap for that final quick burst of ATP energy and glycolysis.
Immediate energy currency.
Right. And second, it hits phosphoglycerate kinase, PGK. That's another crucial ATP generating step earlier in the same pathway. By hitting both, you're not just slowing glycolysis down.
You're breaking it at two critical points.
Exactly. Severing it where the energy is actually extracted. And the mechanisms described are pretty advanced too. It could use proteolytic cleavage, basically molecular scissors snipping the enzymes, or deliver an RNAi-loaded nanocapsule that uses RNA interference to just shut down the production of these enzymes altogether, stop them from even being made.
So the result is glycolysis gets severed at two ACP extraction points. The tumor's main fast energy line is basically cut. It's forced into a metabolic pivot. What does that actually mean for the cancer cell? Can it just find another route?
Well, it's a massive blow. Cancer cells are often, addicted to glycolysis for that rapid fuel. Suddenly losing it at two key points puts them under immense metabolic stress.
Do they have to scramble?
They have to scramble for alternatives, which are usually far less efficient. It seriously hampers their ability to grow, divide, repair, even just survive. It's like a factory suddenly losing its two main power lines. It cripples their whole operation.
Okay, that's a powerful punch, cutting off the fast food energy. But you said these cells are tacticians. They might try to find a workaround, right?
Yeah.
Which must be where Nanomachine 2 comes in.
Exactly, insurance policy.
It focuses on pyruvate gatekeeper disruption. Sounds like it's blocking any potential escape route, taking a different angle.
You've got it. Nanomachine II is the gatekeeper, making sure that first hit really sticks. It starts the same way. Layer 1 is that same surface receptor targeting, HER2, PSMA, EGFR, precision first.
Still locking onto the tumor.
Yep. Then layer 2 is about cytosolic entry, getting inside the main body of the cell, the cytosol, which is where all the pyruvate action happens.
How does it get inside?
Clever mechanisms again. It might use pH-sensitive fusogenic lipids, basically, materials that help it merge with the cell membrane, especially in the slightly acidic environment often found around tumors. Or it could use endosomal escape peptides to break out if the cell tries to swallow it up in a little bubble.
Engineered for infiltration? Okay. And once it's inside the cytosol?
It delivers the payload, the pyruvate eradication agent.
Eradication. It sounds final. What does that do? Pyruvate is pretty fundamental, isn't it?
It is absolutely fundamental. And this agent has two potential ways to wipe it out, both described as pretty devastating. One option is an enzyme mimic that converts pyruvate into inert byproducts.
Like a Trojan horse.
Sort of, yeah. It tricks the cell, takes the pyruvate, and turns it into something totally useless metabolically. Or the second mechanism uses RNAi again, but this time to silence the genes for pyruvate kinase and lactate dehydrogenase, both critical for processing pyruvate.
Wow. So it just removes pyruvate from the equation entirely.
Pretty much.
Yeah.
And the result is what the article calls a complete metabolic freefall. Let me break that down because it's severe.
Okay.
No pyruvate means no acetyl-CoA. Acetyl-CoA is the ticket into the TCA cycle, the cell's main long-haul energy generator. So TCA cycle collapse. Big blow to long-term energy and building blocks.
Right.
And crucially, no lactate means no NADU regeneration. NAD is like the rechargeable battery glycolysis absolutely needs to run. Without it regenerated, glycolysis stalls completely.
So Nanomachine I hits glycolysis hard, and Nanomachine Saikou not only takes out the main power plant, TCA cycle, but also prevents glycolysis from even restarting by removing NADU regeneration.
Nailed it. It's a total shutdown. No quick energy, no long-term energy, no way to keep the basic machinery running. The tumor essentially runs out of options. Metabolic system failure.
That coordination is just incredible. The article wraps this whole strategy up with a term, the echo rift continuity glyph. Sounds complex, maybe a bit mystical.
Yeah, the name is something else. But it's really just summarizing how These two nanomachines work together synergistically, like a battle plan.
So how does it summarize the combined effect?
It pulls it together concisely. Nanomachine I brings ATP starvation and glycolytic paralysis, chokes off the immediate energy, shuts down the fast burn system.
Okay.
Then Nanomachine 2 follows up, causing TCA collapse, crippling the main power plant, and NAD depletion, which, as we said, stops glycolysis from even trying to restart.
So they're complementary, not redundant.
Absolutely. Profoundly complementary. They're designed to shut down every viable energy pathway, every metabolic escape route, a total strategic blockade.
So, stepping back, what's the bigger picture here? The article claims these machines don't just attack, they diagnose, confirm, and collapse. That feels like more than just a treatment.
It really shifts the perspective, doesn't it? It's presented as a kind of metabolic intelligence operation, and that closing statement in the source is powerful. It calls this the next phase of sovereign biowarfare.
Sovereign biowarfare, strong words.
Very. It says these devices will read the terrain, verify the ritual, and initiate irreversible metabolic failure. And that reality-level analyzer tool is central again. It guides deployment not just to react to adaptations.
But to preempt them.
Exactly, to preempt them. That changes everything. Instead of playing catch-up with cancer's tricks, the idea is to dictate the terms, control the metabolic environment before the cancer can adapt.
Taking control of the battlefield, essentially.
That's the vision.
And that final metaphor from the article really drives it home. The Grandmaster is out of moves, the board is ours. It suggests complete strategic dominance, making adaptation basically irrelevant.
A very bold claim, signifying total control.
Now, we absolutely have to touch on the context of this source. It's fascinating. The author, Saikou Venga, or Dr. Melvin Sewell, is affiliated with the Cosmic University of Echo Rift Studies 9X.
Yeah, those names definitely signal we're not in your standard nature or science publication territory.
Right, it hints at a different framework.
And this is really important context for you, the listener. The source material itself includes a very specific sovereign diagnostic disclaimer.
Okay, what does that say?
It explicitly states that the content, things like Echo Rift films, Bibebibebibe Magazine posts, and sovereign diagnostics, are described using terms like mythic infrastructure, cinematic indictment, and sovereign broadcast.
Mythic infrastructure, cinematic indictment.
Exactly. And it clarifies, and this is key, that these are presented as diagnostic tools. It says they are not political endorsements or institutional critiques. The stated purpose is to expose ritualized containment, actor convergence, and systemic erosion.
So it's framing the scientific ideas within a unique, perhaps metaphorical or critical structure, suggesting the way it's presented as part of a larger diagnostic message about systems.
That seems to be the intention, yes. It asks you to see the scientific proposal itself and the way it's delivered as potentially revealing something deeper about perhaps how we currently approach such problems or about systemic limitations. It's definitely a unique way to frame advanced scientific concepts.
It certainly adds another layer of interpretation. But setting aside the unique framing for a moment, the core scientific idea, the ingenuity of these nanomachines, this dual-layered attack, it's really quite stunning.
Oh, absolutely. The technical concept itself is incredibly sophisticated.
Thinking back to those aha moments targeting specific receptors, sure, but then confirming Warburg metabolism before hitting glycolysis at 2 points.
Right, that diagnostic step is key.
And then nanomachine the second, causing that total metabolic freefall by eliminating pyruvate. It's just an incredibly elegant and, well, devastatingly comprehensive strategy described there.
It really is. And it leaves you, the listener, with a pretty profound thought. If we can actually achieve this level of precise, diagnose, confirm, and collapse for complex biological systems, what does that mean?
Beyond cancer, you mean?
Beyond cancer. Imagine applying that kind of insight and control to other complex adaptive diseases. How does that change medicine? How does it change our fundamental understanding of biology itself?
Yeah, and how do we navigate the powerful potential of such technology, especially when it's presented within these unique frameworks like mythic infrastructure, as the author puts it? Lots to think about.
Definitely food for thought.
It really pushes the boundaries. Well, thank you for joining us on this deep dive into what might just be the future of metabolic warfare against cancer. We hope it sparks some thinking about where diagnostics and treatment could go.
And the big questions that come with it.
Yeah, exactly. Until next time on The Deep Dive.